This is a gastrointestinal gram-negative rot tutorial presented by the SchrDbb. We reference Lippincott illustrated review of microbiology by Cynthia Cornilesson, Professor Fisher, and Richard Harvey. So in this tutorial, we're going to continue with gastrointestinal gram-negative rot. So still under the free living bacteria, we have gram-positive and gram-negative. So you can see under the introduction of bacteriology. Now the next, under gram-negative, we have the cocci, we have the enteric rots, and we have the non-enteric rots. So we have already seen the cocci. Is it clear? So the gram-negative cocci. So we have seen with the gram-negative cocci such as Neisseria that we have already done. Now we are going to see enteric rots. So example of enteric rots. So you need to also know that there are enteric cocci also, but enteric cocci are more gram-positive and they are under the chapter known as streptococci that we have already seen before. So now the enteric rot. So enteric rot consists of the Campylobacter. Example of Campylobacter, we have Campylobacter fetus, Campylobacter jejuni. Consists of the Enterobacters. It consists of the Escherichia. Example is Escherichia coli. It consists of the Helicobacter. Example is Helicobacter pylori. Consists of the Klebsiella. Example is Klebsiella pneumoniae, Klebsiella oxytoca. Now, so let's start with the Escherichia coli. So now let's continue again with the classification. This is still under the classification of the enteric cocci. So still under the classification of the enteric rots, we have the we have the Klebsiella, we have the Klebsiella oxytoca, Klebsiella pneumoniae. Still continuing in that, we have the Proteus. So under enteric rots, we have the Proteus, like Proteus mirabilis. We have Providentia. And we also have the Salmonella. Salmonella, example of Salmonella here, we have Salmonella enteritidis, Salmonella typhi, and Salmonella typhimurium. Is it clear? Even Salmonella paratyphi, all that is in this class here. Now we have Serratia. So Serratia is another enteric rot. You have Serratia meracensis. So the next one is Shigella. We have Shigella soni and even Shigella dysentery. Those are examples of Shigella. Vibro, we have Vibro cholerae, we have Vibro parahemolyticus. Yersinia, we have Yersinia pestis, we have Yersinia enterolytica, and we have Yersinia pseudotuberculosis. Is it clear? So all these are enteric rots. Now, for Yersinia pestis is not too much of enteric rot. It's a rot but not too much enteric. So the next one is an electron microscopic view of an Escherichia coli, showing the virulence factor. So the virulence factors of Escherichia coli consist of a lipopolysaccharide, and the antigen of the lipopolysaccharide is going to account for the O antigen. Is it clear? The next virulence factor of Escherichia coli is going to be flagella, and the flagellum is going to account for the H antigen, and the next is the capsule of the Escherichia coli, which is going to account for the K antigen. So all these are the enteric factors of Escherichia coli. Now we need to know that there are different Escherichia coli intestinal infection that occur based on the different microorganisms of Escherichia coli. Now, the Escherichia coli, we have enterotoxigenic Escherichia coli. We have there are 5 main Escherichia coli. The first is enterotoxigenic Escherichia coli. Second is enteropathogenic Escherichia coli. Third is hemorrhagic Escherichia coli. The fourth is entero-invasive Escherichia coli, and the fifth is entero-aggregative Escherichia coli. So these are the abbreviations: the ETEC, the EPEC, EHEC, EEC, and the EIEC. These are the different abbreviations. Now, what are the syndromes each of them produce? Enterotoxigenic Escherichia coli only produce watery diarrhea. Now, enteropathogenic Escherichia coli produces Hawaiian diarrhea of long duration, mostly in infants and often in developing countries. Now, enterohemorrhagic Escherichia coli is going to produce bloody diarrhea, hemorrhagic colitis, is going to produce hemolytic uremic syndrome, enterohemorrhagic Escherichia coli. Now, enteroinvasive Escherichia coli is going to produce bloody diarrhea only, and then enteroaggregative Escherichia coli is going to produce persistent watery diarrhea intrusion and patient infected with HIV. Now, what are the therapy useful in each of them? Antibiotics are useful in toxigenic. Antibiotic may be useful in pathogenic. Avoid antibiotic in hemorrhagic because it has possible risk of potentiating the hemolytic uremic syndrome. Is it clear? We don't use antibiotic in case of enterohemorrhagic because you are going to potentiate the hemolytic uremic syndrome. The question is, why are you going to potentiate a hemolytic uremic syndrome? You potentiate hemolytic uremic syndrome because the toxins which are produced by Escherichia coli are mostly endotoxin. So if you use antibiotic to kill the microorganism, you are going to cause more of the release of the endotoxin when the cells are going to be killed. Is it clear? So you prevent them to be killed by just rehydrating the patient and preventing the use of antibiotics. Now, when you have enteroinvasive, just rehydrate and correct the electrolyte abnormality. And when you have enteroaggregative, you also rehydrate and correct the electrolyte abnormality. So those are the different things to visualize here. So these are the different microorganisms. Now, the next diagram are the action of Escherichia coli toxins. Is it clear? So there are two main toxins. The major toxins that you have, we have the two major toxins. We have the heat libel toxin, that is the LT, libel, L for libel and T for toxin, and then we also have the ST, which is the heat stable toxins. Is it clear? So those are the two main toxins that are produced by the Escherichia coli. So let's start with number one. So how do the heat libel toxin act? So we have the heat libel toxin, means that the heat is capable of destroying that toxin. So the heat libel toxin is going to bind to a receptor and activate the adenylate cyclase. Is it clear? So it binds to the receptor, the adenylate cyclase is going to activate it. So it's going to convert ATP to produce CMP, and CMP now is going to result down to the release of the chloride ion, potassium, hydrogen carbonate, sodium ion, and water. That makes the diarrhea. Is it clear? So that's the adenylate cyclase going to produce the CMP, cyclic AMP. That's going to cause elevated level of CMP, now cause the activation of secretion of ions and water into the intestinal domain. So it just act like the same like Vibrio cholerae toxin. The Vibrio cholerae toxin is still going to activate the CMP. That's also going to result to the release of these ions and water into the intestinal domain. That's going to cause the watery diarrhea. So that is involved there. So the clinical significance, you need to know that the different diseases, the extraintestinal diseases that are being caused by Escherichia coli, you have urinary tract infection, and Escherichia coli is the most common cause of urinary tract infection, including cystitis and pyelonephritis. Is it clear? Now, it also causes neonatal meningitis. Is it clear? So based on the capsular antigen, so just is going to cause neonatal meningitis, and it can also cause a nosocomial or hospital-acquired infection. So now these are based on the Escherichia coli. Now, after Escherichia coli, we now need to know the specific characteristics, the summary of Escherichia coli. So it needs to know that they are short rods, they are facultative anaerobes. Is it clear? They are anaerobes, but they are not complete anaerobes. They are facultative anaerobes, meaning that they are both anaerobes and aerobes. They ferment glucose. Is it clear? They ferment glucose. Most strains ferment lactose. So since they are anaerobes, they are mostly involved with fermentation. Ferment glucose, they ferment lactose, they are catalase positive, they are oxidase negative, and the culture on the McConkey agar. So that's where the culture of Escherichia coli is going to be. So the culture of Escherichia coli is going to be on the McConkey agar, and then you can see here that they are short rods and they are also gram negative. Gram-positive microorganisms are going to be purplish or violet when you are placing them on gram stain, while gram-negative organisms are going to be more pinkish because of the sotranine stain, which is going to make them to be pinkish. All this is going to be seen in the introduction in bacteriology. So you can just pause the video and then go back to the introduction of bacteriology and see all this for 1 hour. So the next one is Escherichia coli. So these are the antibiotics used for the different Escherichia coli infection. For urinary tract infection, you use first line is ciprofloxacin, second line is imidacloprid sulfamethoxazole in urinary tract infection caused by Escherichia coli. The first line is ciprofloxacin, the second line is imidacloprid sulfamethoxazole. Now, the next is local and systemic disease. Now, if you have a bacteremia caused by Escherichia coli, the first line you can use ampicillin, or you can use cefotaxime, or you can use an aminoglycoside, or you can still use ciprofloxacin, you can use trimethoprim sulfamethoxazole. All these can be used as empiric therapy for the first line of Escherichia coli. Now, meningitis in infants, usually infants, you don't give etherinjection cephalosporin, mostly you give cefotaxime in infants, because all of these have mostly side effect in children except for ampicillin, but usually in infants you give cefotaxime. Now, the next microorganism we are going to continue with Salmonella infection causing enteric fever, so like typhoid fever. So you need to know that what this is the lumen of the gut. What's the life cycle of Salmonella? It starts by the Salmonella microorganism is going to be ingested, and then the Salmonella is going to enter the small intestine cell by endocytosis. Is it clear? So later on, now you have macrophages that are going to enter. Now you have Salmonella is going to pass through the endothelial cells to the submucosa, where they are going to be taken up by the macrophages. So the macrophages are going to uptake the Salmonella particles. Then later on, the macrophages are going to carry the Salmonella to the reticuloendothelial system, where the bacteria are going to multiply intracellularly. Though they are still free-living microorganisms, they mostly divide intracellularly. Is it clear? Causing the lymphoid hyperplasia and hypertrophy. So that's why the patients that have Salmonella infection are going to have lymphadenopathy, that are swollen lymph nodes, and they are also going to have splenomegaly and hepatomegaly. So the Salmonella now is going to re-enter the bowel via the liver. So it first starts by infecting the reticuloendothelial system, then it re-enter the bowel via the liver and then the gallbladder. It's now re-enter the bowel via the liver and the gallbladder. That's why initially when you want to diagnose Salmonella typhus based on the clinical symptoms, the clinical symptoms of Salmonella typhus is first starting with a fever, and also starts with lymphadenopathies. The patient has fever and lymphadenopathies with hepatomegaly and splenomegaly. He has systemic symptoms before he has gastrointestinal symptoms, because the Salmonella typhus is going to be released into the bowels later on. Is it clear? So if a patient presents with headache, he presents with fever, he presents with tachypnea, tachycardia, and lymphadenopathies, before later on he has abdominal pain, diarrhea, and vomiting, you can diagnose easily typhoid fever caused by Salmonella typhi. Is it clear? So you can see these are the different disorders caused by Salmonella. You have gastroenteritis, is it clear, which is also called salmonellosis. We have the enteric or the typhoid fever. So what is the general characteristic of Salmonella species? Salmonella species are short flagellated rods, so it means that they can move. Since they are flagellated, just like Escherichia coli, they can move. They are facultative anaerobes. Is it clear? So they are facultative anaerobes, meaning that they can ferment glucose. Is it clear? So they do not ferment lactose. They are catalase positive, they are oxidase negative, and they culture on McConkey agar. Usually, if you are fermenting a substance, that is, you are anaerobic and you are not going to have oxidase. So that's the general characteristics. Oxidase is mostly used by microorganisms that are going to use oxygen for their respiration. Is it clear? You're not going to have oxidase if you ferment glucose, if you are an anaerobe. Is it clear? Generally, that's a principle you can put in your mind. So still, just like Escherichia coli can be cultured on the McConkey agar. So this is where it's cultured, and this is the pattern of growth on the McConkey agar, which is different from Escherichia coli. Is it clear? Now you can visualize here, you see that there are many flagella that you can visualize for Salmonella typhi. Salmonella typhi is still gram-negative rod, but you are going to see many flagella. But for Escherichia coli, you only have one flagella that is going to be seen, one flagellum, sorry. Now the serovar, so Salmonella typhi have two main serovar. You have Salmonella typhi serovar, a variant which is the Enteritidis variant, and a variant which is the Typhimurium variant. The Enteritidis variant is the one that is going to cause enterocolitis, gastroenteritis, and we have also the serovar typhi. So these are the different serovariants of the Salmonella. So the serovariant Enteritidis and the serovariant Typhimurium are the ones that are going to cause enterocolitis, that's gastroenteritis, or even food poisoning. So antibiotics are not normally used except in immunocompromised patients and the spread of infection all through the body. But now, if you have a serovariant typhi, is it clear, which is going to cause typhoid fever or paratyphoid fever, that's when you use now antibiotics like ceftriaxone or ciprofloxacin. Now, this is a micrograph showing the S-shaped cells of the Campylobacter jejuni. So Campylobacter are more S-shaped cells, so as you can see here. So these are the characteristic form of the foodborne illnesses. Now, bacteria that may cause food poisoning due to a preformed toxins, what are they? The first one is Bacillus cereus. Is going to cause also food poisoning from a preformed toxin. We have Clostridium botulinum, Clostridium perfringens, and we have Staphylococcus aureus. All of them, they are due to food and preformed toxins. So because the toxins are injected preformed, no microbial growth within the host is required, and symptoms are rapid. It means that the symptom can occur even 2 to 12 hours after you eat. Immediately you eat food and then you have vomiting, abdominal pain, just know that it's food poisoning because of the preformed toxin. And in those cases, you just need to reallay the patient and then to reassure the patient and to use even antiemetics. There is no need to use in this case antibiotics. Is it clear? So bacteria that may cause foodborne illnesses after food is ingested. So that those bacteria are the ones that are causing now infections. You have Campylobacter jejuni, you have Escherichia coli, you have Salmonella species, you have Listeria monocytogenes, Listeria Shigella species, and Vibrio cholerae. So these ones, because the microbial growth occur within the host, it is required, the symptoms are going to be slowly progressing and usually occur after at least 24 hours after eating the food. Is it clear? So these are the general characteristics of the Campylobacter jejuni. They said that Campylobacter jejuni is curved or spiral or S-shaped rods. Is it clear? They have a single polar flagellum, meaning they are motile. So resulting to a characteristic darting motion. Is it clear? So they are microaerophilic. They also use oxygen. They do not ferment carbohydrate. You have to know that if you are an aero, you are not going to ferment a carbohydrate. Is it clear? Fermentation is mostly used for anaerobes, and you can see that in the introduction of bacteriology. So for you to understand it better, you just pause and go back to introduction of bacteriology and look all those videos. Now the next is culture on the selective medium. It can also be involved in culture and of selective medium. So the selective medium that you use to culture Campylobacter species are the blood agar containing antibiotics to inhibit the growth of fecal flora. So this is Campylobacter jejuni. As you can see, they are S-shaped or they are curve shaped. on Campylobacter jejuni, and this is example of a medium, like the medium that can be used, a Preston selective medium for Campylobacter jejuni, and you see specific growths occurring there. Now for Campylobacter jejuni, there are different diseases involved in Campylobacter jejuni. You have acute enteritis, you have traveler's disease, and you have pseudoappendicitis. Pseudoappendicitis can be found in case of Campylobacter jejuni, or it can still be found in case of Yersinia enterolytica. So you're going to see that later. So in all these cases, acute enteritis, traveler's diarrhea, or pseudoappendicitis, you use erythromycin and ciprofloxacin. Is it clear? Now, the next, you have mechanism of Shigella infection causing diarrhea. So now this is the life cycle of Shigella. So for life cycle of Shigella, you have ingestion of Shigella. So the Shigella is going to be ingested still at the level of the large intestine. In the case of Salmonella, the cells that were ingesting the Salmonella were mostly at the level of the small intestine. Here you have ingestion, and then the Shigella is going to enter the large intestinal wall and also the rectal wall via endocytosis. Is it clear? So now in step 2, the Shigella are going to escape the endocytic vesicles, and then they are going to multiply inside the cells. So they are going to multiply inside the cells, and they are protected from macrophages. So you see that in this case, Shigella do not enter macrophages like the Salmonella. So the Shigella just continue to live inside the enterocyte. Is it clear? Now the Shigella now are going to invade now the neighboring enterocyte, and they are going to try to evade from the macrophages, because the macrophages are going to kill them. Now the next one is mucosal abscess form as the cell dies. So when the enterocyte now die, you have an abscess formation. Is it clear? And you have a diarrhea containing pus, blood, mucus, and painful abdominal cramping, because the Shigella infects the enterocyte. So general characteristic of Shigella is that they are non-motile, they are non-encapsulated, they cannot form lactose, they cannot ferment lactose, and most strains do not produce gas in mixed acid fermentation. So they can also do fermentation, and if they do fermentation, it means that they are anaerobic. Is it clear? The culture of the Shigella is done on the hectoing agar. That's general characteristic. So this is hectoing agar that has been shown there, and this is the pattern of growth of Shigella on hectoing agar. On gram stain, you can see that there are rods which are more cluster. Is it clear? On gram stain, you need to know that the gram stain, gram negative, is going to be pinkish color. As we have said, gram negative is pinkish color, while gram positive is purplish color. The purplish color comes because of the iodine stain, while the pinkish color comes because of the sustranin stain. So just pause and go back to the introduction of bacteriology that we have done in Science Video Makers, and then you look those videos. So then this is a specific characteristic of Shigella sonnei. Shigella sonnei is the one that is going to cause bacillary dysentery or shigellosis. Is it clear? Or sometimes you may call it Shigella dysentery. Is it clear? So the antibiotic that you use for Shigella is going to be azithromycin, that is a macrolide, or you use a ciprofloxacin. Now, the next microorganism I'm going to visualize is Vibrio cholerae in cholera. So let's see the life cycle. So you need to know that the Vibrio cholerae is not the one which is involved in the infection. Is it clear? It is the toxin which is involved in the disease. So we have the cholera toxin, which is also called the cholera gen. So the cholera toxin has different groups. It has the A and it has the B group, as you can see here. So you have the B subunit is made up of 5 subunits, and the A subunit you only have 2 A subunits. Is it clear? It's going to bind now. So this is the GM ganglioside receptor. Is a receptor at which is going to bind. Is it clear? When it binds to that receptor, you have the GS protein close, and you have the inactive adenylate cyclase. So now, when it binds as such, you have the release. The A subunit is going to enter the cell membrane. It's going to activate the GS protein, which is going to in turn activate the adenylate cyclase. So when the adenylyl cyclase is now going to be activated, the active adenylyl cyclase now is going to convert now ATP to produce the CMP plus the inorganic phosphate. Is it clear? So now we have the adenylyl cyclase going to produce now elevated level of the CMP, and then CMP is now going to result to the secretion of the ions and the water molecules, as you can see here. So this is how is the action of the cholera toxin. So now the next one is virus cholerae species. For virus cholerae species, this is the general characteristic. You need to know that it is short, curved, or rod shape. For viral cholerae, so they are curved and they are rod shaped or comma shaped. So they are rapidly motile as a result of a single polar flagella. They are facultative anaerobes. That's why immediately somebody dies, cholera is going to die with that person within 24 hours because they are going to be exposed to air. They are facultative anaerobe. Growth on many virus strains require a stimulated. So they are facultative anaerobe, meaning that they are not going to immediately die. They can also grow in presence of air. So that's why every time somebody dies from cholera, you must make sure that that body of the patient should be carefully taken care, handled by a health personnel. Else the patient who is dead may transmit cholera to another person. Is it clear? So we have growth of many viral strains is required and stimulated by sodium chloride. Now you need to know that though you still need sodium chloride for the stimulation of the viral cholerae strain, the culture of the viral cholerae species is done on the McConkey agar. So this is viral cholerae, which is done by scanning electron micrograph. You can see how they are common in shape. And now to treat viral cholerae or cholera is mostly fluid rehydration, because the toxins are the one which is mostly involved here, and you have a small treatment therapy with antibiotics such as doxycycline or ciprofloxacin as first line. So now the next disorder is going to be Yersinia. And then Yersinia has different pathogenesis that we are not going to speak about here. You can pause and see the pathogenesis of Yersinia. It consists of Yersinia enterotica, Yersinia pestis, and Yersinia pseudotuberculosis. Now for Yersinia species, you need to know that it is motile, it is non-capsular, it is found in contaminated water supply, unpasteurized milk, and also contaminated food. So the gram stain of Yersinia enterotica is shown here. For Yersinia enterotica, is also called Yersiniosis. It's going to cause gastroenteritis and can also cause septicemia. So example of drugs that can be used in this case can be ciprofloxacin and trimethoprim sulfa methoxazole. Now the next disease, the next microorganism we're going to speak about is Helicobacter pylori in the gastric pit. So the Helicobacter pylori in gastric pit can be seen when you are doing a biopsy of the gastric pit. You're going to see gram-negative rods here like that. So you can see gram-negative rods. So what is the pathogenesis of Helicobacter pylori? What happens is that the first step is that the Helicobacter pylori is going to penetrate the mucous layer lining of the stomach epithelium, and it is the only microorganism that grows in the stomach epithelium because of the extreme acidic pH of the stomach epithelium. Is it clear? So it's going to be attracted to the chemotactic substance hemine and urea. So it's going to be penetrate the mucosal lining of stomach epithelium and attracted to the chemotactic substances hemine and urea. Is it clear? The next, these are, you can see Helicobacter pylori all through here. Helicobacter pylori. Now Helicobacter pylori now recruits and activate inflammatory cells. You're going to have inflammatory cells are going to try to come to destroy the Helicobacter pylori. They also release the urease that cleave the urea and produce the ammonia and neutralizes the stomach acid at the vicinity. Is it clear? So this is how the Helicobacter pylori strive to grow inside the stomach mucosa. They grow in the stomach mucosa by producing one enzyme called urease. When urease is going to produce, it's going to convert the urea, which is normally present in blood, into ammonia and carbon dioxide, and the ammonia is a base that can neutralize the acid in the stomach. That's one method. Another method is that the inflammatory products, since there is an antigen, an abnormal antigen inside the stomach, the Helicobacter pylori is going to recruit the inflammatory cells that are going to lie around the mucosal membrane where Helicobacter pylori is going to stay, and when they lie there, they are going to make sure that the acid gastric juice does not touch them. Is it clear? So the inflammatory cell can also protect as a coating against as an indirect coating against the Helicobacter pylori, because they are trying to kill those Helicobacter pylori. The next is Helicobacter pylori cytotoxin and ammonia produced by the urease can cause now the destruction of the mucosal cells. Is it clear? So the ammonia produced and the cytotoxin produced by the Helicobacter pylori is now going to cause the destruction of the cell and also the destruction of the connective tissue of the stomach wall. So the stomach acid now is going to act now directly to the connective tissue, because there is no mucosal cell to protect the stomach wall. Since there is no mucosal cell to protect the stomach wall, now the patient is going to complain now of pains at the level of the chest, because the stomach is being burned out by the acid. Now these are the specific characteristics of Helicobacter pylori. It is curved, it is spiral, it's a curve or a spiral rod. They have multiple polar flagella. Is it clear? Which give the organism a rapid corkscrew motility. They are urease positive. They are this culture on selective medium containing antibodies. So this is Helicobacter pylori culture. So on a selective medium containing antibody, and this is a gram staining. Gram staining shows a gram usually in vitro. It is a frequently gram positive. Is it clear? So when you see a purplish appearance, you may think that it is a gram positive microorganism, but usually Helicobacter pylori is more considered as a gram negative rot. Is it clear? So the next thing is Helicobacter pylori here. You have this is the treatment of acute gastritis. Treatment of acute gastritis, you use three main drugs. You first use a proton pump inhibitor, then you use amoxicillin and clarithromycin as a tritherapy. Now, you can also use a tetratherapy of proton pump inhibitor plus amoxicillin plus clarithromycin plus a metronidazole or tinidazole. Is it clear? Now, this clarithromycin here is a macrolide. Is it clear? So you use a penicillin plus a macrolide plus an imidazole. So all this is used as a tetratherapy. That one is used for a tetratherapy for Helicobacter pylori. Or if you want to use the second line of Helicobacter pylori, when you don't want to use amoxicillin, you are going to use bismuth sulfate. It's still going to kill the Helicobacter pylori. Now we are now going to move on to Enterobacteriaceae family. So this is a micrograph showing the rod-shaped Klebsiella species. So this is rod-shaped Klebsiella. So these are the example of the Enterobacteriaceae family. So in the Enterobacteriaceae family, other Enterobacteriaceae family consists of the Klebsiella. It consists of the Enterobacters. You have Enterobacters, which are example of Enterobacteriaceae family. You have Klebsiella. We have Serratia, we have Proteus, we have Providencia, we have Morganella. Is it clear? So these are the different disorders that you have visualized here in cases of this disease. So from here we can say thanks for your kind attention, and please don't forget to like and subscribe for our channel Science Video Makers. Thanks very much.